let this be a normal field trip? With a friend? No way! Magic School Bus!
Good morning, class. How did everyone get to school today?
I walked.
My mom drove me.
I biked today.
I bust!
So today we will learn about the cell's modes of transport, cell motility. Cell motility has a whole bunch of different functions such as development, immune system defense, and locomotion. Whereas some cells use actin and myosin to facilitate these movements, others use small hair-like structures such as cilia and flagella. All of these transport structures are seen in unicellular eukaryotes such as amoebas and also multicellular eukaryotes such as us humans.
Ms. Frizzle, how does ciliary and flagellar mediated transport differ from the actin and myosin dependent transport we saw last week?
Well, Hosein, there's only one way to find out. Let's go on a field trip to the bus.
Ms. Frizzle, what's that, a furry cell?
Actually, what you are seeing is not a singular cell, but rather a small ciliated marine organism called a Trichoplax adherens. It is known as the simple motile animal. Even though it lacks a nervous system and a muscular structure, it has the capacity to coordinate locomotion and other complex behaviors.
But if it has no nervous system, how is it able to coordinate its movements?
While trichoplaxes are lined by monociliated epithelial cells which convert chemical energy into mechanical work by the beating pattern of its cilia, ciliary flocking, which is the action of single cilia multiplied to form collective and coherent movements, explains how trichoplax can coordinate its locomotion in the absence of a nervous system. This can be described as a sort of self-assembly.
According to my research, trichoplax can crawl on surfaces since their cilia exhibit what is called a walking gait. Ciliary walking patterns arise from the interplay between cilia's interactions with the surface they are crawling on and their oscillating beating motion. So it's cilia's altering pattern of adhering to a surface and propelling off of it that enables them to move forward.
That's right, great job. Despite their simplicity, trichoplax exhibit complex ciliary dynamics, therefore making them an ideal model system to study ciliary locomotion and motility.
But Ms. Frizzle, how can this research regarding a very simple animal help us better understand ourselves?
Well, the cilia that you see allowing the trichoplax to move work through a highly conserved mechanism that is present in many cells found within our own bodies. Why don't we take a look?
I should have stayed home.
Here's one example. The bronchi of the lungs are lined with mucus that trap microorganisms and debris. Under this mucus is cilia that moves the mucus out of the respiratory system to be coughed out or swallowed.
Wow, they're beating in unison, just like the trichoplax cilia do.
That's right, the mechanism for the cilia to beat in rhythm is actually very similar to the trichoplax. Cilia tend to receive signals from their neighboring cilia, which allows for coordination in beating. Let's move up and take a look into the central nervous system.
Not again!
Whoa, look, there it is again. Their cilia are moving something around.
That's right, even in here, cilia play a vital role in maintaining the health of an organ system. In the subarachnoid space, cerebrospinal fluid flows in order to bring nutrients to and waste from the brain. However, the CSF needs to continually flow in order to achieve this, which is exactly what the cilia are doing.
Are there any other examples of cilia?
Of course, there are a lot of different examples aside from what we've explored, like in the ear and in the fallopian tube.
But the magic bus is running out of magic juice, so we have to head back to class for now.
Cruising on down Main Street, you're relaxed and feeling good. Next thing that you know, you see octopus in the neighborhood, surfing on a sound wave, swinging.